in this video, we'll be looking at cell transport. So we'll be looking at 3 different transport mechanisms that are used in moving things in and out of the cell in different scenarios. Largely, they could be classified into 2 different types of transport, as you can see on the diagram right now, and we'll go through them one at a time. First of all, let's start on what we call a passive transport. So passive transport means that these are transport mechanisms that doesn't require energy to actually happen. So they happen quite naturally, and that means that these are mechanisms where the chemicals are moving from an area of high concentration to an area of low concentration, or to simplify, we say the chemicals move down the concentration gradient in the 2 passive transport mechanisms. We'll look at the first one, which is diffusion. So diffusion is probably one of the most simplistic ways of chemicals moving. These could be chemicals moving across a space, or it could be moving into or out of a cell. And there are 2 main points that we need to include when defining what diffusion is. So diffusion is defined as the net movement of particles down the concentration gradient. The word net refers to overall. So when it comes to a scenario, let's say we've got 2 spaces, and here I've got particles, lots and lots of different particles there. And then on the right hand side, I've got particles, but less of them. So generally speaking, that the particles are free to move about randomly in all sorts of direction. They could go from left to right or from right to left. But the thing is, in this case, generally speaking, the particles will be moving from the left towards the right, which is what we mean by from an area of high concentration to an area of low concentration. So this is what we mean as diffusion. Or we can say it down the concentration gradient because it's going from high to low. So diffusion is the net movement of particles down the concentration gradient. Now, a lot of things could move by diffusion, and we're going to look at a particular example, which is gas exchange. So for example, let's say in human beings, we've got red blood cells, or any cell basically that requires oxygen to survive and to function, because oxygen is needed for aerobic respiration, which releases energy. So in this case, we say that oxygen would diffuse into cells and carbon dioxide can diffuse out of cells. Right, so these 2 gases move in and out of cells by diffusion. Other examples obviously could include, let's say, plant gas exchange, which is about, again, same with oxygen and carbon dioxide, moving in and out of the leaf through the stomata, and again, that is diffusion as well. Another common question that is often asked is, what are the factors that affect the rate of diffusion, and specifically how do we increase it? Because diffusion happens in a lot of different places. About, like I said, gas exchange. It could be about any other chemicals that are moving in and out of cells, or any exchange surfaces such as the alveoli and the villi as well. So actually there are a few common things that these exchange surfaces have that allow a faster diffusion rate. So one of the key ones is about having a higher surface area to volume ratio. A lot of different cells and tissues and organs would do this by having longer extensions or have a slightly curved surface. So for example, red blood cells have a biconcave shape, which increases surface area to volume ratio, allowing faster diffusion of gases into and out of the red blood cell. Small intestines would have villi on them. Again, it's about increasing the surface area, allowing for faster diffusion or faster chemical movement. The lungs have lots and lots of alveoli, which increases surface area to volume ratio, allowing for faster diffusion of the oxygen into the bloodstream and faster rate of carbon dioxide diffusing out of the bloodstream. So actually, surface area to volume ratio is a common theme in exchange surfaces. Another common one is about these exchange surfaces having a thinner membrane, and the key thing here, which people often forget, is about explaining why having a thinner membrane leads to faster diffusion, and actually it's because they provide a shorter diffusion distance. And the name is quite self-explanatory. The chemicals don't have to go through that long of a distance, so obviously diffusion happens quicker. Having a higher temperature is another reason for that, because the higher the temperature, then that leads to a higher kinetic energy in the particles. So Ke stands for kinetic energy. That kinetic energy is the energy found in moving objects. So if you heat up the particles, then the particles have more kinetic energy, meaning they are vibrating more and they're moving around more. Then obviously the faster they move, the faster the diffusion would happen, because diffusion is the movement of particles. The final one is about having a steeper concentration gradient or maintaining a steep concentration gradient. So this could be in animals, for example. This is usually done by having an extensive blood network, or having lots and lots of capillaries wrapping around certain exchange surfaces. And the idea is that once the chemicals move into the bloodstream, they're very quickly carried away, so that you're keeping the difference in the concentration between the surfaces. So let's say, for example, here I've got an area that has a couple of these chemicals. So 1, 2, 3, 4, 5, 6, 7, so 7 of them. And on that side, I've only got 2, versus another situation when I've got, let's say, again, 1, 2, 3, 4, 5, 6, 7 of the particles there. But then I've got 1, 2, 3, 4, 5 on the right-hand side. Then obviously the particles are free to move around however they like, but because of the difference in the concentration, diffusion would happen a lot quicker here than on the top bit here than the one on the bottom. So it would still happen, but obviously this one is a lot faster because of the steeper concentration gradient, and this one will happen a lot slower. So this section here summarizes what diffusion is, which is a passive mechanism of transport, meaning it doesn't need energy, and it's the net movement of particles down the concentration gradient. And there are several factors that would affect the rate of diffusion. Now the next one that we're going to look at is osmosis. Osmosis is also another example of a passive transport, meaning it doesn't require energy, but osmosis is specifically the movement of water molecules. Sometimes people say it's the diffusion of water, and actually that is quite accurate. So osmosis is defined in 3 particular points. Osmosis is the net movement of water molecules down the water concentration gradient through a partially permeable membrane. Now again, remember the word net, which means the overall, because again it is a passive movement, so the water molecules are free to move across the membrane from in any direction, but generally speaking it's going to go from an area with lots of water to an area of little water. The other thing to make a note of is that the difference between the definitions of osmosis and diffusion is that you must specify that it's going down the water concentration gradient, and that is key. Because if you just set the concentration gradient, it could mean the concentration of any solute that is in the solution. So if we look at this particular example, you can see that on both the left-hand side and right-hand side of the membrane, we've got 4 solute particles, let's say sugar particles or salt particles. But you can see on the right-hand side, there's a lot more water molecules than the left-hand side. So in comparison, we can say that the right-hand side is what we call dilute. It's got a lot more water, whereas on the left-hand side, this is more concentrated. It's kind of like when you're making squash, for example, or Ribena, you get the concentrated version, and then you have to dilute it with water by adding water to it to make it taste less strong. So in this case, because osmosis is the net movement of water molecules down the water concentration gradient, so in some sense that the dilute one would have a lot higher water concentration. And on the left-hand side, the concentrated one, there's a lot less water there. So that is why osmosis would actually occur from the dilute one, where there's a high water concentration area, to a low water concentration area. So even though the left-hand side is called concentrated, but that means that there's less water. So in fact, it's got lower water concentration gradient. So that's why it's important that when you are defining osmosis, make sure you say down the water concentration gradient. And it has to happen through a partially permeable membrane. That's one of the prerequisites for osmosis to occur. So a classic example of osmosis is in the root hair cells. So in the soil, there'll be lots and lots of water, and the root hair cells would be in comparison having less water, so the water will move in by osmosis. I mean, obviously water moves in and out of many cells, but the root hair cell is a very specific example where we can talk about osmosis happening. Now, one of the trickiest parts in osmosis is about understanding how osmosis actually affect animals and plant cells, depending on what kind of solution that they are submerged in. There are mainly three ways to describe solutions, depending on if they've got more water inside in the solutions or more water inside the cell. So the first one we're going to look at is a solution called a hypotonic solution. So hypo means that the solution that the cell is in has a lot more water than inside the cell cytoplasm. Because the hypotonic means there's more water outside the cell, the water would move into the cell by osmosis. Now remember, whenever you're explaining this one in exam questions, make sure you say the direction of the water movement and that this happens by osmosis. Even if you can't say the word hypotonic, as long as you can describe the water concentration difference inside the cell and out of the cell, and then also describe the water movement, that is absolutely fine. So because the hypotonic solutions have a lot more water than the cytoplasm, water moves into the cell by osmosis, and by osmosis is 1 mark in exam questions. Now thinking about animal cells, if there is a lot of water moving into the cell actually burst. Right? The cell would burst, so it basically just pops like that. So the cell essentially dies. On the other hand, we got a specific type of solution which is called hypertonic solution. The way to remember this is that hyper, think about someone who's very hyper, probably because they had a sugar rush. So hypertonic is referring to a solution that has a lot more sugar or any solute than inside the cell. So in comparison, if they've got more solute in the solution, that means they've got less water in the solution compared to the cell. So therefore, the water would actually leave the cell by osmosis. So it's actually the exact opposite to hypotonic solutions. Now, because the water is leaving the cell by osmosis in a hypotonic solution, for an animal cell, it just becomes shriveled. It becomes shriveled up. So it kind of lost its shape and it becomes smaller, and it just doesn't really quite retain the same sort of, let's say, circular round shape that it might have. So animal cells in these two different solutions would either burst or shriveled. So the best thing is actually having the right amount of water in the bloodstream to make sure that your cells don't burst or shrivel up. So in plant cells, let's say if the plant cells are found in hypotonic and hypertonic solutions, the water movement is still the same. So in hypotonic solution, again, the water is still moving into the cell because there's a lot more water outside. In a plant cell, that is actually quite a good thing because plant cells do like to have lots and lots of water in it. So we describe the cell as turgid. Right. So in a turgid situation, keeping in mind that the plant cells have a cell wall. So if we draw it out, it will look something like this. So imagine that I've got the nucleus there, and I've got a massive, large, permanent vacuole, because a lot of the water would actually move into the large permanent vacuole to store up those extra water. So in the case of turgid, the cell membrane is actually pushed right up against the cell wall. So it's not just touching it, it's pushed up right against it. Now, sometimes we actually think about, okay, what if there's a bit of water, but not too much and not too little, and the water concentration is actually the same inside and out of the cell? That's what we call that as an isotonic solution. So iso means the same. This means the water concentration is exactly the same in the cytoplasm and out of the cell, and in this case, the osmosis will not actually occur. So no osmosis because there's no net movement of water into and out of the cell. In this case, the plant cell is described as flaccid. Right, flaccid. In this case, the cell shape still looks the same. It's still got the right shape, so with the cell wall there as well. The difference though is that the cell membrane is now still touching the cell wall, but just about touching the cell wall. So you might see a little bit and it's kind of might be leaving it a little bit, but it's still generally speaking touching the cell wall. And the large permanent vacuole may be still quite large, but not massively big compared to the turgid state. So they still have the same size and the same shape of the cell because of the cell wall, but it's going to be, the cell membrane is going to detach from the cell wall just a tiny bit. Whereas in hypertonic solution, the water is leaving the cell by osmosis, and the plant cell is described as plasmolysed. So in this case, it still has the same shape because the cell wall is there to maintain the shape. But this time, because there's so little water left inside the cell membrane almost completely detaches from the cell wall, like that. So it can detach utterly, and maybe the vacuole will be a lot smaller. It's still there, but it's a lot smaller. So keep in mind that the only reason that the plant cell isn't bursting if it's hypotonic solution and it's still able to keep its shape in hypertonic solution is because the cell wall maintains the shape of the cell and it provides the cell shape and support. Whereas in animal cells, if we look over here, because animal cells do not have a cell wall, therefore there's nothing stopping the cell from bursting if it's in hypotonic solution, and it completely loses its shape in hypertonic solution because the cell wall isn't present. So this is how animal cells and plant cells would behave in different types of solution because of osmosis. It is really important that you remember the direction of water movement. Make sure that you mention the word that they do this by osmosis. And a typical question they would ask is the osmosis PAG, or the required practical, about putting the potato cylinder in different concentrations of, let's say, sugar solutions. And make sure that you think about how we can use these words and the concept of hypo and hypertonic solutions and isotonic solutions to figure out the concentration of potatoes. So now we're all done with the passive transport mechanisms. Now we're going to look at some of the active, or one particular active transport. Now remember that passive means that it does not require energy, and that's why active means that it needs energy to actually occur. And the name is actually quite direct, and you would have heard it before, of course. This is active transport. Now, active transport, as the name implies, requires energy, and it's moving chemicals in a different direction. So active transport is the movement of particles against the concentration gradient using energy and carrier proteins. So you can see that we're not talking about net movement anymore, because net movement implies that the particles are allowed to freely move across the two different directions. But here, actually, we are specifically directing the particles to move in a particular direction here. So the classic thing is that they do have carrier proteins which act as gates on the cell membrane. So kind of like this. So let's say, for example, we've got some particles here, and this is the difference in the concentration. So you can see on the right hand side there's a lot more of them, and on the left hand side there's only 3. But despite that, I still want to make sure that I absorb all of these particles from the left to the right hand side, so from the area of low concentration to an ever higher concentration. So the idea is that this gate that I have here is going to be able to change its shape. Right, so energy goes in to change its shape and to pump and move these particles from an area of low to high concentration. And that's what active transport is. Now, obviously, when you think about some examples where this happens, and usually this means that the particles that we're trying to move into towards the high concentration area means that we need these particles. These particles are important and essential for our functioning. So there are 2 examples that I'll talk about, one in animal and one in plants. The one in animal, a common one, is talking about the small intestine. The small intestine is responsible for absorbing nutrients. And keeping in mind that actually, even though our cells already, or our bloodstream already has a lot of nutrients in it, it's got glucose and it's got lots of mineral ions, etc. But that doesn't mean that we want to stop absorbing them, because we need all of the nutrients that we can get. So the small intestine would have lots and lots of mitochondria in their cells, meaning that the mitochondria can release the energy for active transport to occur, absorbing these nutrients. An example for plant cells would be root hair cells, and that's a very, very common one. So root hair cells in the roots, in the soil, there will be lots and lots of mineral ions in the soil because of fertilizers or various other reasons. And because even though, like I said, same as the concept of small intestine, the root hair cells already have lots of minerals in them, but it doesn't mean that the plant doesn't want more. So they would keep absorbing the minerals from the soil by using active transport. So this sort of, in this topic, they quite like to link to other things such as explaining the adaptations of specialized cells. So you can talk about how the cells in the small intestine are specialized to have lots of mitochondria so that they can release more energy for active transport. And the same for root hair cells, of course. So there you have it. These are the 3 transport mechanisms. To summarize, we've got generally 2 types. We've got active and passive transport, that depending on if they need energy or not. Active transport uses energy to move chemicals against the concentration gradient by using the energy to change the shape of the carrier protein. For passive transport, 2 types would be diffusion and osmosis. Diffusion is basically the net movement of particles down the concentration gradient. And osmosis is a form of diffusion, specifically diffusion of water. So we need to say down the water concentration gradient when talking about osmosis. And it has to happen through a partially permeable membrane. And keep in mind how animal cells and plant cells would behave depending on what concentration of solutions they are in. And this is everything you need to know about cell transport.